The study of proteomics gives us information about the quantity, about the presence, about the interaction, and the sequence of the different proteins which are present. And in order to study this, we have to perform, or we have to take the help of computers, because data is extremely huge. And the study which is done, that is through the bioinformatics. Bioinformatics, that the information which is present in the biology or in the biological composition. In this module, I will try to present an overview of different systems, different softwares which are used to study the bioinformatics. Now, this is understood that as our databases about the proteins and the proteomics, that enlarges and that swells with billions of bases of sequences from the human and other genome, and countless protein structures which are discovered, protein-protein interaction. So when the data is so huge, a crucial problem may be to access and manipulate all these data. That how to access this data, how to use this data beneficially. And this is done by bioinformatics. So what bioinformatics is defined as? That the bioinformatics, it systemizes the data and solve the problem of easy access to this data available on the computer or available on various databases. Scientists, they have prepared different databases, and the practitioner of bioinformatics, as understand or as indicated, that they must understand both biology and computerized data processing to make use of maximum information. So in this case, a hybrid between the computer technology, IT, and the biology, molecular biology, is required, which is known as the operational bioinformatics or bioinformatic scientist, which is there. Now, different databases which are presently available, they are, number one, BLAST. That is a program that searches a database for DNA or protein sequence. You know, when there is a DNA, there is a protein sequence. So DNA or protein sequences, which is similar to a sequence of interest. So if we are interested in a sequence of different protein, different DNA, then the BLAST is the program, and it shows how the two sequences line up. How the two sequences, they line up, they are similar or they are different from that. Another program is GRAIL. Now this program, it identifies gene in a database. You see, it identifies the gene which are present in a database, whereas the BLAST, that determines just a database for DNA protein sequence. It gives us a sequence which is similar to of our interest. GRAIL, it gives us it identifies the gene which are present in that database. Now, the two types of databases that have been established. You see, the GRAIL is for the database. So it has two types of databases has been established which are available. One is generalized databases. They are general. And the second is the specialized database, which is a specialized for certain genes, for certain things. Now, generalized databases, as the name indicates, they include DNA and protein sequences from all organisms, because that's a general one. So that includes a huge sequences of DNA and all that. Now, two generalized databases for DNA sequences which are available. One is the GenBank and the second one is EMBL. So one is GenBank and then EMBL. Whereas the Swissprot is a generalized protein sequence database. The one earlier, they are for DNA, the GenBank. And if you are looking for a protein sequence, you have to access the Swissprot, that is a database. Now, specialized databases, now these databases, they deal with a particular organism because they are specialized. For example, FlyBase is a database of the genome of the fruitfly, Drosophila, and specialized database, one can search it for the genetic maps of the genes, DNA sequences, and other information which is available through these databases. A similar important site is a WormBase that provides the same kind of data for the nematode C. elegans. So you see that there are so many databases available. The databases are available for DNA sequences, for the protein sequences, for different species, and for all the species which we can access on these programs, and that is the closal information which is present in them is available to us through the bioinformatics. So what we do with those that information, that can help us to understand, to construct, and to make the diagnosis properly and precisely.